Last time we talked about the different ways that viruses can get into hosts. And now for the next two sessions, we're going to talk about how the host responds in defense. So we're going to do some immunology. Now that, of course, could occupy an entire course, and it does here. There is a course on immunology. So we have to restrict what we do. We can't do all of immunology. And my focus is going to be virus-centric, how it applies to viruses, and in particular how viruses counter defenses of the host. Because, as you'll see, host defenses are great, but obviously viruses have to get around them, otherwise they would never infect. And that's going to be my focus, to tell you how viruses get around these defenses. And today we're going to start with what I call intrinsic and innate defenses. Next time we will do adaptive defenses. So what is the difference? This is the virologist's view of the immune response. Brick walls, a series of brick walls. And each one is slightly different. So today we are talking about intrinsic and innate defenses, but they're not actually the first defenses. The first defenses are anatomical and chemical that are always present, like skin. I consider that a defense. Mucus, of course, bathing mucosal surfaces, it's a barrier. Saliva, stomach acid, tears, etc. Defensins, you have antimicrobial polypeptides on your skin that are antibacterial largely, and some antiviral. They're produced by skin cells as well as your resident microbiome, so they're important. All right, so that's continuous. Then we have intrinsic, which we'll talk about today, and these include autophagy, apoptosis, microRNAs, CRISPRs, and many others, but we'll restrict to some examples. And then we have innate immunity. And in contrast to the first two I've told you about, innate immunity takes minutes to hours to turn on. This is relatively quick, but it's not always present as the first two are. And this includes cells and complement and cytokines, which we'll talk about today. And then next time we'll devote ourselves to the adaptive or acquired immune responses, T cells and B cells, which takes time. Takes a couple of weeks to develop. So everything else is what is there before adaptive immunity, because it takes too long. If you just had adaptive immunity, it wouldn't be good, because you wouldn't have time to get rid of pathogens. So each defense is a wall, and viruses get over the first, the second, and third, and if they last, they encounter acquired immunity, and if that doesn't work, then you have a serious disease. So let's start with, again, a more definition of these host defenses. Intrinsic, as I said, which we'll talk about today, is always present in the uninfected cell, and we'll talk about some of these mechanisms today. The innate immune response is induced by infection, but it takes minutes to hours for it to appear. And the adaptive immune system is tailored to the pathogen, and that's why it takes a couple of weeks. So the other two defenses are not tailored. They're general defenses, the intrinsic and the innate defenses. So let's talk about some intrinsic defenses. First is RNA interference. So in this defense, a virus is infecting its cell. If this is a virus with an RNA genome, the RNA gets into the cell, and it's recognized by a nuclease, an endonuclease in the cell called dicer, which is named so because it chops up the RNA into short double-stranded pieces, about 21 nucleotides or so. And then one strand of that duplex, again, this is part of the viral RNA genome, it's been chopped up. One strand becomes part or binds to the RISC complex, the RNA-induced silencing complex, and this contains a number of different proteins, including Argonaute. And this complex will then find a target viral RNA that's complementary to this short sequence, and it will guide the RISC complex to it, and it will chop up the RNA. So it is anti-RNA defense based on the sequence of the RNA. The RNA comes in, it's chopped up, and those pieces then direct the RISC complex to chop up more genomes, so this can clear the infection. Now, we know this happens in plants, We know this happens in vertebrates. In fact, that's where it was first discovered. We're not sure about mammals. There's a controversy in the field over whether RNA interference is actually an antiviral mechanism in mammals or not. And we may not need it because we have developed protein-based immunity. This is RNA-based immunity. And mammals may not need it because we have adaptive responses that are quite sophisticated. So right now, we're not sure. However, in plants and invertebrates, when viruses infect them, the viruses encode countermeasures. So I'll have on almost every slide today this word countermeasure, because if viral genomes didn't encode countermeasures, they would never replicate. They would be cleared by these wonderful defenses. So viral genomes encode antagonists of RNA binding proteins that are part of this pathway, the nucleases and so forth. We don't need to go into them in detail, but they exist. Here's an example of intrinsic immunity that exists in mammals. And this is a protein called APOBEC3, and it works, the example I'm going to show you is how it works for HIV-1. APOBEC3 is apolipoprotein B mRNA editing catalytic polypeptide. You don't need to know that, but you should be curious about what abbreviations mean, right? APOBEC, there are many different kinds of APOBECs in us, and they have very interesting functions, but one of them has the function of being a deaminase, a cytosine deaminase. What does that mean? Okay, so in HIV, this protein, A3G, APOBEC3G, would be incorporated into the virus particle as it buds off. There it is, the orange ovals. And then when this virus particle infects a new cell, right, the DNA is reverse transcribed, the RNA is reverse transcribed. And that's where APOBEC would work. And let's look at this little box at the bottom. The viral RNA, there in green, minus strand DNA synthesis. And then the APOBEC deaminates the DNA, so it changes C's to U's. You remove the amine from the C, it becomes a U. And now when you replicate or you copy the DNA and you make the second strand, you make mutations all over the place. You make hundreds of mutations because there are hundreds of C's in the DNA, and that mutates the virus to lose its infectivity. This has been called a weapon of mass deamination, WMD. Why does HIV exist with this? Well, because HIV has a small protein encoded in its genome called VIF, VIF, the blue one up there in the upper left, which targets APOBEC to the proteasome, and it gets degraded in infected cells. And we know this because you can make HIV lacking VIF, and in fact, that's what's shown at the bottom here, VIF minus, and that's the only condition under which APOBEC gets incorporated into the HIV particles if you take out VIF. With VIF, HIV degrades APOBEC so it never gets incorporated, and that's why we have HIV as a problem. So it's a countermeasure to this lovely intrinsic defense. And that's what I mean when I say that without countermeasures, viruses wouldn't be around. Another really important one that we are currently learning more and more about in terms of its importance is epigenetic silencing. What this means is when DNA, foreign DNA comes in the nucleus wants no part of it. You can imagine that's not a good thing to have DNA coming in all the time. And it happens all the time, not just by viruses, but cells pick up DNA and it makes its way to the nucleus. So the DNA is silenced epigenetically. And all that means is that the chromatin is compacted. DNA, if it comes in the nucleus and it's not already wrapped around chromatin, it will be wrapped around chromatin and then silenced by methylation of the chromatin, methylation of the DNA, deacetylation of the histones, many mechanisms. So here we have compacted chromatin because it's been chemically modified, and it can't be transcribed because it's compacted. But here we've acetylated the histone tails on the right. That opens up the chromatin, and it makes it transcriptionally accessible. So when many viral DNAs come into the nucleus, they're silenced by these mechanisms. And they're often put into what are called PML bodies. These are nuclei of DNA virus-infected cells, and they're stained with an antibody to one of the protein components of these bodies, which are basically silencing places. The DNA is routed into them, and it's modified so that it's its chromatin is compacted and it's silenced. Of course, virus genomes encode countermeasures. For example, human cytomegalovirus encodes a protein that leads to degradation of this cell protein called DAX, which is needed for histone deacetylation. Other viruses encode proteins that affect the protein localization of PML or synthesis, so silencing is reversed basically in these genomes. Now, just today, I put in the next slide, which you will not have, because this is a very cool new discovery of epigenetic silencing that has to do with retroviruses. And I just learned this. My colleague Steve Goff at Columbia works on this, and this is from his paper, which was published recently. And if you're interested, we did a TWIV on this, which we call Quiet Please, because we're silencing the genome. That's his joke, not mine. Remember, retroviruses have RNA genomes. They come in the cell, the genome is reverse transcribed to make DNA, which then goes in the nucleus. This is just DNA coming in the nucleus, so what do you think is going to happen? It's silenced. And that's what he's discovered. He discovered that the linear DNA, there are actually circular DNAs that are made as well. We didn't talk about that. These are all silenced by methylation of the histones, by deacetylation, because the cell doesn't want this genome to be expressed. So what's the countermeasure for this virus? It integrates into the host cell genome, into loose chromatin. Remember, I told you retroviruses like to integrate into areas which are being transcribed. So the escape here is that the DNA integrates in all the repressive marks. So the histone modifications, methylation, acetylation, we call those marks. They can be written and read, so they're written by enzymes and read by other enzymes. And the integration of the retroviral DNA into the genome removes all the repression, and that's why the genome can be expressed. So I think it's a brilliant way of countering without having to encode a protein that counters it. Other viruses do, of course, but this is a nice example that you don't have to encode a protein to escape this epigenetic silencing. So once again, as the DNA come in to the nucleus, they're wrapped around histones, and then histone deacetylases and methylation work to silence it. But the integration removes the silencing. All right, so then another intrinsic defense is apoptosis or apoptosis. You can say it however you want. I've been beaten into saying apoptosis by my British colleagues. But I used to say apop, but you can have all kinds of arguments why it's apoptosis, or the other, helicopter is not helicoder, right? It's a helicopter, but still they want me to say apoptosis. So that P is supposed to be silent. Big argument. Apoptosis is programmed cell death, and it has roles in normal development. When you need to get rid of cells during development, they undergo programmed cell death, in which the cell basically breaks up, the DNA breaks up, the cytosol comes off in blebs, and the blebs can be picked up by macrophages. Now, if this is a virus-infected cell, virus infection can stimulate apoptosis. We'll actually talk a little bit about that later when we talk about viral-induced cancers. And it's an intrinsic defense because the cell is giving up, I'm infected, but let's save the rest of the organism, so we'll undergo apoptosis. You'll see later today that these apoptotic bodies are picked up by macrophages and dendritic cells as a way of saying, do we have foreign antigens around? And so that's what I mean by apoptosis is monitored by sentinel cells. A macrophage and a dendritic cell are sentinels that circulate throughout all of our tissues, and they pick up these apoptotic bodies and they ask, is there anything foreign in them? And if so, they will mount a response. So apoptosis is a intrinsic defense. And of course, viruses have to counter this, otherwise they would never get anywhere. And in particular, the DNA viruses with longer replication cycles that take days to weeks to replicate, they have to counter apoptosis because apoptosis can happen in hours and they would never replicate. Some of the smaller RNA viruses that replicate quickly actually stimulate apoptosis as a way of getting out of the cell. They inhibit it initially, and then when they've made particles that need to get out, they stimulate it. But all these DNA viruses, you can see adenovirus, herpes viruses, pox viruses, they're all DNA viruses. They encode a variety of inhibitors of apoptosis. Apoptosis has many targets where you can interfere proteins like BCL2 caspases, et cetera, et cetera. There are multiple targets. So each of these viral proteins can inhibit apoptosis at these different steps. And again, you don't need to know all these steps, but you do need to know that viral genomes encode inhibitors of apoptosis. Otherwise, they wouldn't be able to finish their reproductive cycles. Then, of course, there's CRISPR-Cas defenses, which is found in most archaea and half of bacteria. You know, this is something everybody on the street probably knows about because CRISPR systems have been repurposed for gene editing now. And soon 3 people will get Nobel Prizes for this work because it's really important. But what it is a defense. It's an ancient defense mechanism for these microbes. In one iteration, if a phage, a virus of bacteria, infects a bacteria, it injects its DNA into the cell, of course, which will go on to reproduce, but the host encodes nucleases, which will chop it up and integrate small pieces of the phage genome into the host DNA. And a surviving bacteria will then have an array of these bacterial phage sequences in the bacterial genome. And the CRISPR array is derived just by that, clustered regularly interspaced short palindromic repeats. And then the next time, these things are always constitutively transcribed, these arrays, to make CRISPR RNAs. And so if a phage then infected that cell, those CRISPR RNAs, together with a CRISPR-associated nuclease like Cas9, would chop up the incoming phage DNA. And so this is really a history of infection. You can now match phages with their hosts by looking at the CRISPR arrays that are present in the host cell. So this kind of defense works for viruses or any incoming DNA, plasmids or any environmental DNA that would get into the host cell. And it's extensive. There's more than one nuclease and so forth. It's incredibly extensive. And of course, this nuclease plus the CRISPR RNA has been repurposed so that we can now edit genes very specifically. But it is a defense that is present in these hosts. And it's an intrinsic defense because it's always present. This first question is about intrinsic defenses. They're always present. Which of the following are included? Antibodies, T cells, epigenetic, silencing, skin, or mucus, which is an intrinsic defense. So the answer is C, epigenetic silencing is an intrinsic defense. Antibodies and T cells are adaptive. It's the last defense that takes a couple of weeks to induce. Skin is one of those earlier ones, anatomical defenses. They're always present, but it's not called intrinsic defenses. So mucus is the same as skin. It's just a matter of what I call it. And in fact, I will bring you back to the first slide to clarify that, the wall. See, anatomical and chemical are skin and all these things, and intrinsic are down there. And that includes epigenetic silencing, which we talked about. All right, so that's intrinsic. I want to talk about innate defenses now for the rest of the time, and that's that third wall there. This is a defense that requires minutes to hours to be activated by infection. It's composed of 5 components: cytokines, sentinel cells, which include dendritic cells, macrophages, and NK cells, and complement. 1, 2, 3, 4, 5, okay? 1, 2, 3, 4, 5. And this innate defense can stop infection, but as you will see later today and next time, it can also collaborate with the adaptive defense and say, I can't handle this infection, you need to take over. And we'll talk about how that happens. How was this discovered? So for years, people knew there were innate defenses, but we didn't understand how the host distinguished foreign things from self, because the innate system detects proteins and nucleic acids, and a protein is a protein, a nucleic acid is a nucleic acid, or is it? Anyway, this begins back in 1980. 2 scientists in Germany are actually studying development of Drosophila, fruit flies. And they're making mutations First time you could do this, making mutations throughout the fly genome and looking at the effect on development. And they found a gene which, when mutated, it altered the dorsal ventral axis in the flies. And legend has it that Christiane Nusslein-Volhard was looking in the microscope at the phenotype, and she called over her collaborator, Wieschausch, and she exclaimed in German, "Das war ja toll," which is like far out crazy. Look at this phenotype. And so this gene was called the toll gene, right there, because of that. Well, that's what the story says. Anyway, they got the Nobel Prize for this really important discovery. So this is a gene that was initially determined to be involved in fly development, and it turned out it had a role later on. It was found out to have a role in immunity of the fly to microbes. And then people found toll genes in mammals. And it turned out that those were part of the sensing system for the innate immune system. So we called them toll-like receptors in mammals based on the original toll gene in flies. Now there are a bunch of toll-like receptors. These are called pattern recognition receptors. They recognize something that's foreign, that's not self, and they initiate an innate immune response. So here on the top left is a schematic of what these look like. These are transmembrane proteins. They're on the plasma membrane or sometimes in the endosomal membrane. Here's the original toll gene from Drosophila on the right. And the left, TLR1, toll-like receptor 1 through 10. And you can see they all have a similar structure. There is a cytosolic domain. It's called the TIR domain, which stands for toll IL-1 receptor domain. And then there are a series of leucine-rich repeats that constitute the extracellular part of these molecules. Now, the structure of a few of these have been solved, and you can see on the lower left the structure of the extracellular domain of TLR3. You can see the leucine. You can't see that they're leucines, but you can see repeats, right? Obviously, there's a short sequence that is repeated to form the curved structure of this protein. Those are leucine-rich repeats. They're full of leucine. This particular molecule binds RNA. That's an RNA molecule, a double-stranded RNA molecule in there, and it's being recognized, which causes dimerization. This is a common theme, that recognition of the ligand causes dimerization. And so on the right is a table that shows what kinds of macromolecular patterns are recognized by these toll-like receptors. And you can see there's a whole variety of things that range from bacteria to viral components. For example, triacylated lipopeptides, peptidoglycan and lipoteichoic acid, components of the outer wall of bacteria, lipopolysaccharide, flagellin is recognized by TLR5. And then we have nucleic acids. We have single-stranded RNA and double-stranded DNA, single-stranded RNA there, even CG dinucleotides. So very specific components. These signal to the innate system that there's a foreign entity present, like a virus or so. Now you may say, how does nucleic acid signify foreign? Well, you'll see in a moment. So that's toll-like receptors. And since they were discovered, we've also discovered cytosolic sensor proteins. So here on this diagram, we have a toll-like receptor on the plasma membrane, and then we have a variety of TLRs in the endosome, because of course, when viruses enter cells, many of them go through an endosome, and that's a perfect place to have a sensor for, say, nucleic acid, because these TLRs can sense a variety of nucleic acids, you can see there, and the cell is concluding that because this is in an endosome, it's probably foreign. So if the TLR recognizes it, we're going to initiate an antiviral response. We've also discovered cytosolic pattern recognition receptors. Actually, PKR, which we talked about the other day, which recognizes double-stranded RNA, is a recognition receptor. It leads to the inhibition of protein synthesis, as we talked about, by phosphorylation of VIF2 alpha. There's another one called RIG-I in the cytosol, which recognizes RNA. This recognizes RNA with 5' phosphates. So that in the cell, you never have RNA with 5' phosphates floating around. And so it's recognized as foreign. Cell RNAs are usually capped. There's another cytosolic sensor called MDA5, which recognizes long double-stranded RNA, which is also something you don't find floating around the cell. It's usually viral. So when these sensors detect their ligand, whether it's in the endosome or in the cytosol, that starts a signal transduction pathway, which ends up resulting in the expression of specific genes in the nucleus, and genes for cytokines of a variety of types, which we will talk about in some detail. And these mRNAs come out of the nucleus, they're translated, and then the proteins are secreted, and they affect other cells as well as the cell in which they're made. So you can see that the signaling pathways are very simply shown here. If RIG-I senses RNA, it goes through a signaling pathway that leads to this protein, IRF3, getting phosphorylated. It's a transcription factor. It then goes in the nucleus and turns on specific gene expression. The same thing for the TLRs. IRF7 is a transcription factor. It's phosphorylated in response to sensing the ligands for these TLRs, goes in the nucleus, turns on genes. NF-kappa B, same thing. So foreign nucleic acids are recognized by cytosolic proteins like RIG-I or membrane-bound TLRs. And they initiate the production of cytokines, which we will see are antiviral. DNA can also be sensed. Here is a pathway that was discovered relatively recently, where we have a DNA virus coming in to the cytoplasm, and some DNA gets out into the cytosol. And there shouldn't be DNA floating around in the cytoplasm. It should be confined to the nucleus. And so this DNA is sensed by a protein called cGAS. And this is an enzyme which, when it's bound by DNA, it synthesizes from ATP and GTP a cyclic molecule called cyclic GAMP. It's just shown here to show you what's going on. So here's an AMP molecule and a GMP, and the phosphates are simply joined in a cyclic fashion. That's done by cGAS, and so cGAMP is made, and then that binds to a protein on the ER called STING. STING then goes through the Golgi and turns on a couple of kinases that phosphorylate. IRF3, which is a transcription factor, goes in the nucleus, or activates NF-kappa B, which goes in the nucleus, and that then turns on the genes of cytokines that have antiviral properties. So again, just like for the RNA, here we're sensing DNA by an unusual pathway, but in the end you're sensing DNA, sending transcription factors into the nucleus, which turn on the synthesis of cytokines. These sensing pathways also exist in the nucleus, because many viruses put their DNA in the nucleus. Of course, viruses encode modulators of these pathways. They have to, otherwise they would not be able to successfully reproduce. And in fact, every virus has to encode some kind of antagonist of all these sensing pathways. This is just one, but I could show you a slide with all of them and it would be full of text because every pathway would be inhibited. So here is a cell infected with a herpes virus, Epstein-Barr virus, and this encodes a protein called BPLF1, which is the blue oval there, that antagonizes the detection of viral proteins by TLR2 on the cell surface. So TLR2 can detect viral proteins, and when it does, it initiates a signaling cascade that results here in the entry of NF-kappa B into the nucleus, turning on the genes encoding pro-inflammatory cytokines, which they, again, have antiviral activities. This BPLF interferes with that in a number of ways. For example, this signaling pathway requires the addition of ubiquitin to components of the pathway, those little green U's, those ubiquitin molecules that are needed for their activity, and BPLF1 interferes with that, so these proteins cannot participate in the signaling. In addition, it blocks the proteasomal degradation of the I-kappa beta alpha, B alpha subunit of NF-kappa B. When this is removed from NF-kappa B, then NF-kappa B can go in the nucleus. So there I-kappa B alpha is bound. It remains in the cytosol. When it's taken off, and it's taken off because it's degraded, phosphorylated and degraded, then NF-kappa B goes in the nucleus, but BPLF1 can block the degradation, which causes NF-kappa B to remain in the cytosol. So those are just three examples of how a viral protein can antagonize this pathway. They can antagonize at every step of these rather complicated pathways that I really haven't shown you. Next question is, which of the following allow the innate immune system to distinguish microbes from self? cytoplasmic helicases and toll-like receptors, antibodies, apoptosis, APOBEC, all of the above. So the answer is A, cytoplasmic helicases and TLRs. These allow the innate immune system to distinguish microbes from self. The toll-like receptors can distinguish viral proteins, viral nucleic acids, and the same with the helicases, generally just nucleic acids. None of the others are correct. Antibodies and apoptosis and APOBEC do not distinguish self from non-self. Now, among the products induced when these TLRs and the helicases sense something foreign, those are cytokines. And one of the very important cytokines is interferon. So we'll spend a little time talking about interferons. This was discovered in 1957 when these two investigators found that if you added influenza virus that had been inactivated to chicken cells, the chicken cells will produce a soluble substance that, if you add to other cells, can block their infection. So they say it interfered with infection. It didn't even require infectious influenza virus. It could be done with non-infectious virus. So they called it interferon. And people worked with this for many years until we understood that it is a product of the innate immune system, and it's one of the cytokines that are turned on when a TLR, for example, senses something foreign. So they're produced by virus-infected cells. They're also made by sentinels. Macrophages and dendritic cells will make interferon, even if they're not infected. And what happens is shown at the bottom here. We have an epithelial cell sheet with our basement membrane. So up on top, if this is a respiratory tract, that would be the airway. Viruses are infecting these cells. They're sensed by TLRs and helicases. The cells then produce cytokines. Those are the red dots. And they're diffusible molecules, so they float down to other cells and they float below the basement membrane. And the dendritic cells are patrolling constantly. They're all over us. They're below our epithelial sheets. They're in our major organs, in our skin, and they will sense the production of cytokines and home in on those cells and make more interferon. They can also pick up dead and dying cells, pieces of cells released by apoptosis. They will be picked up by these dendritic cells, and that stimulates them to make more interferon, even though they're not infected, because they have receptors that can sense these foreign proteins. There are three major types of interferons, type 1, type 2, and type 3, and each has a distinct receptor on the cell surface, on the plasma membrane, that engages the interferon. So type 1 interferons, there are 13 different types of type 1. That's the purple oval there. That's the interferon molecule, and it's binding a interferon receptor, which is composed of two different chains. And in the same way that type 2 interferon, or interferon gamma, recognizes or binds a specific receptor, as does type 3, which is also interferon lambda. That's the red oval there. So interferons are soluble proteins produced in cells that come out of the cell, then they bind plasma membrane receptors, and that's how they exert their activity. Interferon proteins themselves, alpha, type 1, type 2, type 3, they are not antiviral by themselves. If you add them to virus particles, it will have no effect on the virus particle. Rather, they induce many, many thousands of proteins, which in turn have antiviral activity. So here's what happens. You get a cell infected. Within hours, interferon alpha beta is turned on. Get a rapid burst of it, and it's actually down by 10 hours, so it's a very quick acting thing. This interferon alpha beta comes out of the cell. It binds to interferon receptors on the cell surface, as I just showed you. So here we have in the upper right some interferon produced by sensing in the infected cell. Remember, TLRs and helicases sense the presence of something foreign. The cell produces interferon. It binds to its receptor, and that binding then initiates another signaling pathway, distinct from the one involved in sensing. It involves phosphorylation and dimerization of this protein, which will eventually go in the nucleus and act as a transcription factor. So two STAT proteins join with IRF9, they're phosphorylated, and then they go in the nucleus. They bind to a specific sequence in a promoter of a subset of genes called interferon-stimulated genes, and they turn on the transcription of those genes. And those are the cytokine genes that are induced by interferon. So interferon is a cytokine, and it in turn also induces other cytokine synthesis. And these interferon-stimulated genes, we call them ISGs. There are over 1,000 of them, maybe more, and they are the ones that have antiviral activity. And we're going to go through each one of them now. Each one of the 1,000, okay? Actually, we don't know how most of them work. We only know about 20 or 30 of them. So you're off the hook, okay? But there's one thing I want to tell you about these promoters, which is so cool. And so there is a promoter being bound by this transcription factor, and it's turning on these genes. This promoter, many of those come from retroviruses. We have taken retrovirus LTRs, which have promoters in them, and used them for interferon responses. That's what's shown here. And endogenous retrovirus LTRs regulate the interferon response. So what has happened is an ancient retrovirus many, many years ago infected a germ cell, became endogenized. Here's the endogenous retrovirus. And over time, this underwent recombination and put the LTR next to a gene that is involved in interferon responses. And so basically, these LTRs are what respond to the transcription factor that comes in the nucleus as a result of interferon binding to the cell surface receptor. Many of them are derived from retrovirus LTRs. 6 to 14% of the mammalian genome has promoters derived from LTRs. You can see they've moved away from the endogenous retrovirus. The LTRs have recombined and moved elsewhere, but it contains a promoter, of course, which has evolved to become responsive to the transcription factor that goes in the nucleus when interferon binds the receptor. And that promoter, I'll go back to it, that promoter, it's called ISRE9 here. The motif, the sequence in that is called a gas motif. And that's what's donated by the retrovirus. And you can see in all these lineages, we have humans and monkeys and rodents and bats, horses, cows, elephants. They all have LTRs driving part of their interferon response. Isn't that amazing that we have co-opted this? So that's a really cool story. But another example of how we have used retroviral elements in this case, to run our defenses. Now let's talk about just a couple of these interferon-stimulated genes. Remember, interferon itself is not antiviral, but it turns on the synthesis of ISGs, interferon-stimulated genes, of which there are over 1,000. And let's talk of just about a couple of them. Here's Tetherin, also called CD137. This is a very cool one. This is a molecule shown on the slide on the left image in blue. It's a plasma membrane protein, which essentially tethers virus particles to the cell surface. Here we're showing HIV being tethered to the cell surface. So they can't butt off so they don't infect other cells. So this is an antiviral protein. It's holding HIV on the cell surface. So you may ask again, just as you did for APOBEC, why do we have HIV if we have Tetherin? Because the virus antagonizes it. And the virus also encodes a small protein called VPU, which binds Tetherin in the endosome and doesn't let it get out to the cell surface, or the membranous vesicle, which would normally transport Tetherin to the cell surface. VPU binds it up, and it doesn't get to the surface, so the virions can butt off. So VPU antagonizes Tetherin at every step of the way. There's antagonism of the innate defenses. Now, if you make an HIV without VPU, that on the right is what an infected cell looks like. The viruses are all stuck on the surface via Tetherin, chains of viruses, because the Tetherin can actually get into the virus particles as well. And the two Tetherins interact, and they hold the viruses to the surface. Look at this. The whole mass of virus particles on the cell surface are butting off, and they can't go anywhere. It's a full subway. You can't get any more particles out, and they're not going to infect other cells. So it's a quite nice antiviral, but the virus encodes an antagonist. Here's another cool one I particularly like. They are called interferon-induced proteins. IFIT1, interferon-induced protein with tricopeptide repeats 1. And this protein has the property of binding to the 5' cap of RNAs that lack 2' O methylation. So here's the 5' end of an mRNA. There's the cap structure, which we touched on previously. At first base of the RNA with the red label, and then we have the cap, and a 5' to 5' linkages with 3 phosphates in between the G and the first base of the RNA. And typically, 3 different positions in this area are methylated. The cap G itself, and then base 1 and base 2. That's 2' O methylation because it's on the oxygen off the ribose ring. And IFIT binds RNAs that lack 2' O methylation because a lot of our RNAs lack 2' O methylation. And so when IFIT1 binds, it prevents the cap binding protein from binding, and so the RNA is not going to be translated. So again, this is an antiviral protein made by the host cell. And on the right is the structure of this protein solved a couple of years ago. You can see in red is the G. This is called M7G. It's just the brown part, the G and the base with the methyl, and that's the red structure that's bound in a pocket inside of this IFIT1 protein. And on the right, you can see a space-filling model where we've cut away the IFIT1, and you can see the M7G in there. That will only bind if it lacks the methyl, the 2 methyl groups, because the methyls are going to block the entry of the cap into this protein if they're present. So any virus that lacks 2' O methylation in its capped mRNAs are going to be bound by this, and their translation will be inhibited. Of course, there are many ways that viruses get around that. Escape from IFIT1. One example is influenza virus, which steals the cap from the host. The host always 2' O methylates those 2 positions. So any capped mRNA from the host is not susceptible to IFIT1. And because influenza virus steals its caps on its mRNAs from the host cell, it's not inhibited by IFIT1. Maybe that's part of why it steals caps, in addition to needing them for primers. The coronaviruses don't have a cap. They have a little protein on the 5' end. In them, the ribosome binds internally, and so IFIT can't bind there. There's no cap to bind to, so they escape IFIT1. We have some viruses that encode a 2' O methylase, these viruses right here. So they methylate it, and they escape IFIT because they have malmethyls, and the IFIT can't recognize the cap. And some viruses actually steal host methylases to methylate the cap and get around it. And finally, some viruses have an RNA structure right near the cap that is so close to the cap that even though the cap is not 2' O methylated, it blocks IFIT1 from binding sterically. So there are a number of ways that viruses get around this, what would otherwise be a really cool way of inhibiting replication. All right, so that's all I'm going to tell you about ISGs. What I want to do now is tell you that interferon is dangerous. Even though it's antiviral, it can be dangerous because it induces the production of many proteins. And most of our cells have interferon receptors, so when you are infected, even a respiratory infection, your respiratory cells are making interferons. They can diffuse throughout your whole body and bind to receptors on any cell, make interferon-stimulated gene products, and they can have deleterious effects, like fever, chills, nausea, and malaise. And every virus infection always makes interferon, and that's why flu-like symptoms are so common. Flu-like symptoms doesn't mean you have influenza. It means that you are making interferon, and that gives you fever, chills, nausea, malaise, muscle aches, all those generalized things that you get when you have flu, which is a respiratory infection. Why are your leg muscles hurting? Virus is not replicating there. It's because interferon is going there and doing things. Why do you feel foggy? Virus is not in your brain, but interferon can get in there and it can turn on ISGs. So you have interferon to blame for those systemic symptoms when you get a virus infection. And that's what I meant the other day when I said a great deal of the signs and symptoms of a virus infection are caused by immune responses. How do interferons limit viral replication? Interferons directly inhibit viral translation. Interferons lyse viral particles. Interferons induce ISGs. Interferons damage cells, none of the above. Therapy for hepatitis C for many years was to give people interferon. And half of the people who got it didn't work. But in the other half, they felt lousy. Because if you've ever taken interferon, you get fever, malaise, nausea, chills. It's a crappy drug to take. So now we have better antivirals for hep C. So the answer is C. Interferons induce ISGs. That is how they limit virus replication. They don't directly inhibit translation. They don't lyse viruses. They don't damage cells. As I said earlier, if you added interferons to viruses, nothing would happen. They have to act in a cell to turn on interferon-stimulated genes. Let's move to some other components of innate responses. The sentinel cells, dendritic cells, macrophages, and natural killer cells. So here we have a dendritic cell on the left, very characteristic, almost neuron-like appearance. An NK cell there on the right. And these are cells that are always moving through you in all of your organs, in your circulation, beneath your skin, beneath your epithelial surfaces in your respiratory and alimentary tracts. They're everywhere. You're full of them, and they're looking for trouble. So that's why we call them sentinels. And there are many, many different kinds of dendritic cells, as you can see in this picture. Here on the lower left are some of them. There are blood dendritic cells specific for blood with different markers. There are tissue-resident dendritic cells. Langerhans are the kind that are in your skin in particular. And then there are other kinds as well. And as I said, these patrol all over your body. So on the top, we see some dendritic cells that are patrolling your epithelial lining of your intestine. And in fact, here's one. This purple one has stuck its process through the space between the cells and is looking out there for foreign antigens. I told you that can happen the other day. These red ones are virus particles, of course, and they're dendritic cells underneath. So they're looking for trouble. They're looking for infected cells. And also our other parts of our body, our tissues. Here we have some dendritic cells in very specific places in epithelia, but your larger organs have them as well. They're basically looking for a virus-infected cell. And how would they detect that? Well, if an infected cell is making cytokines because infection's been sensed, these dendritic cells will detect those cytokines. And they will take pieces of the dead cells or viral proteins that are coming out of that dying cell, and they will bring them to the lymph node. And that's what's shown here. These dendritic cells are going into the lymph node where they have these packets of proteins, and they will present them to lymphocytes and say, is this foreign? And if they're not, they go about their business again. But if they are foreign, then the lymphocytes, which will eventually form the adaptive response, make antibodies and cytotoxic T lymphocytes. And so that's an important part of what dendritic cells do. And here's a wonderful picture where we have, this is taken in a lymph node, and these are blood vessels in red, and the lymphocytes are stained blue, and there is a dendritic cell in green snuggling up to a lymphocyte. It's probably got some peptides that it's showing to the lymphocyte, and if they're foreign, the lymphocyte will get activated and begin to differentiate into a virus-specific lymphocyte. So here, that's summarized in this slide. We have our epithelial sheet being infected. The cells are making cytokines. Remember, part of these cytokines are interferons that are going to induce ISGs and inhibit virus replication in these epithelia. But the cytokines will also attract dendritic cells, and they will pick up the cytokines. The dendritic cells will make more interferon often and leave it behind to try and inhibit replication. But more typically, they will pick up viral proteins or nucleic acid released from infected cells. These dendritic cells then mature and they become typical looking dendritic cells. They go into the lymph node where they're then going to interact with the adaptive response. And we'll talk about that more next time. So here's a dendritic cell before and after activation. Here on the left, an unactivated dendritic cell. This is the kind that's patrolling your tissues. It has all kinds of receptors. It's got toll-like receptors on it. It makes endosomes. And here we have some MHC class II molecules holding viral peptides in orange. So these cells can take up viral proteins that are released from dying cells and chop them up and put small peptides into MHC2. We'll talk more about that in detail next time. These have all sorts of receptors. As I said, they can make interferon. But when they do pick up viral proteins, and again, they're stimulated by viral proteins, cytokines, pieces of dead and dying cells. These become activated. They migrate to the lymph node. And then there they can interact with lymphocytes. And here you see it's interacting with a T cell. It has now presented these viral peptides on MHC class II. So these are viral peptides from the infected cell that have been released when the infected cell died. They're processed by the dendritic cell. They're displayed on the surface. And that is how they're presented to the T cell. So the MHC2 on the DC presents the peptide to the T cell receptor on the T cell. There's an exchange of cytokines. There are other receptor ligand interactions as well. But if that peptide is foreign, that will activate the T cell, or it could be a B cell as well, and then they will go on to mature. If the peptide is not foreign, it ends there, no activation of the lymphocyte, and the dendritic cell leaves. So this is how we link the innate with the adaptive responses through these sentinel cells like DCs and macrophages. They detect early events and bring in packets of viral proteins into the lymph node to interact with the adaptive response. The last member of this sentinel is the NK cell, which is shown here. And NK cells patrol also, and they're looking for absence of MHC2 molecules on cells. And here's a target cell. Many viruses, when they infect cells, they remove MHC molecules from the surface, so those cells won't be infected. Because as we'll see next time, it's by these MHC molecules displaying viral peptides that a T cell will recognize this cell as infected and kill it. So many virus infections downregulate MHC2. And so an NK cell recognizes two receptors on the cell surface, an activating receptor and an inhibitory receptor, which engages MHC. Typically, this interaction between the inhibitory and MHC inhibits the other, and so there's no killing of this cell. But if MHC is absent, the negative influence is gone, the activating receptor will lead to killing of the cell. So the idea here is the NK is not actually detecting an infected cell, it's simply detecting an absence of MHC molecules on the surface, which is typically caused by a virus infection. So this is another component of the innate response that happens early on. And yes, there are viral modulators of NK cells. Viral proteins can interfere at all these steps and fool, for example, the NK cell. They can put up MHC homologs on the cell surface and fool the NK cell into thinking that it's not infected, do all kinds of things like that. There's another part of the innate response, which is a soluble set of proteins found in the blood. And this is called complement. It's a series of many different proteins which basically recognize patterns on viruses or bacteria and help bridge the adaptive and innate responses. And there are a couple of different components to complement. One is called the classical pathway, but it involves antibodies. So here's a microbe. It could be a bacterium or a virus. And you're making antibodies. So antibodies are made later on in infection, 2 weeks later. And those antibodies will bind the virus or the bacterium. There's a complement protein called C1, which will then bind the antibodies, and then it activates a series of other complement proteins. All these C2s and 4s and so forth are complement components that act in a cascade. And so, for example, recognition of a foreign microbe by antibodies will trigger a complement cascade that can lead to the production of proteins that are pro-inflammatory. They will recruit cells into the infected area. Another set of proteins will lead to the formation of a pore in the membrane that will actually lyse. It could be a bacterium, it will lyse it, or if it's an envelope virus, it will cause lysis as well. It's called the membrane attack component. Other parts of the complement pathway can help to coat viruses and bacteria with antibodies so they're taken up by macrophages. It's called opsonization. And some other components of this pathway can recognize carbohydrate components on viruses or bacteria. So this is an important pathway for bridging innate and adaptive responses. If you have previously seen a virus infection, you have some antibodies circulating already in your blood, so you don't have to wait 2 weeks for those to arise after a second infection, and the complement system can help clear viruses early on by using those antibodies. And of course, yes, there are viral modulators of complement. So what we've painted so far is a picture of virus infection leading to what we call the inflammatory response, the production of cytokines that cause inflammation. So what is inflammation? Well, if you go online and look it up, you get pictures like this, which say that inflammation is part of many different diseases like cancer, heart disease, Alzheimer's, pulmonary, arthritis, autoimmune, neurological disease, diabetes. So in all these conditions, there's damage that leads to the production of cytokines that cause inflammation, that recruit immune cells to the area. Of course, what's missing here, they don't have infectious diseases. A huge driver of inflammation is not in this one picture that I found. But if you work out and you have joint pain afterwards, it hurts in your knees or whatever, that's inflammation. It's damage, and it's causing cytokine production and recruitment of immune cells. So when infected cells produce cytokines and chemokines, which we'll talk about in a moment, that leads to inflammation. Inflammation was originally described by Celsus in the 1st century AD. He said inflammation is rubor, dolor, calor, and tumor. Those are 4 signs of inflammation that he defined. That means redness, pain, heat, and swelling. And they are caused by increased blood flow, increased capillary permeability, the influx of phagocytic cells like macrophages and dendritic cells, and tissue damage that may occur as a consequence of all that. So when you feel pain after any of these disease states, including infection, it is typically these 4 signs. And they are caused by these changes that are driven by the production of cytokines and chemokines. And so let's talk a little bit about cytokines in general. We've touched on interferons, but we haven't mentioned any others. There are generally 3 groups of cytokines. We have pro-inflammatory cytokines like IL-1, TNF, etc. These promote white blood cell activation. So they're pro-inflammatory. They lead to a lot of those symptoms of inflammation that I just mentioned. And then, of course, you need to have anti-inflammatory cytokines, which suppress the pro-inflammatory cytokines, which is PICs. You cannot have inflammation go on forever. You have to have it modulated by ourselves. And so these anti-inflammatory cytokines are made, which will dampen the inflammatory response. Otherwise, we get more tissue damage. And then we have chemokines like IL-8, which have a specific role to recruit immune cells to an infected area. So when a dendritic cell is moving to an infected cell, in part it's because chemokines are produced from the infected cells that attract the dendritic cells. Now, as I said before, these cytokines, they function locally initially. At your respiratory epithelium, they're produced to recruit cells, produce interferon to inhibit replication, but they can enter the circulation. They are soluble, and they can go throughout your whole body, and that's why they have global effects. They cause flu-like symptoms because they come out of your infected lung. They can go to your liver. They can go into your brain. The chemokines and cytokines go in your brain, cause fever, fatigue, and sleep. Of course, they also go in the bone marrow. Some of these are colony-stimulating factors that go into the bone marrow and drive hematopoiesis to make more blood cells. Because you have an active infection, you need more than are just circulating. Some of these chemokines and CSFs go in the bone marrow and stimulate the production of cells there as well. So these have global effects. Even though they're intended initially to be local, they have global effects. A localized viral infection produces global effects. Here's an example of how a chemokine works in attracting a neutrophil to infected tissue. Now, chemokines work through signaling pathways just like interferons do. Remember, interferons bind receptors, they initiate signaling, leading to the entry into the nucleus of a transcription factor that turns on the synthesis of mRNAs encoding ISGs. So here we have a similar pathway for a chemokine. There are chemokine receptors on the plasma membrane that bind the chemokine. There's signaling pathways initiated. Transcription factors are phosphorylated, in this case dimerized. They go in the nucleus and they turn on genes that express the activity of the cytokine. And I'll show you what I mean by that in a moment. So here we have an example where we have a neutrophil in the blood vessel. This is a capillary, and here's the endothelial cells. So this is the lumen of the capillary at top, and underneath is the tissue. So this tissue is infected, and this neutrophil is sensing chemokines that are being produced as a consequence of that infection, and it will eventually move across the endothelial barrier and get into the tissues where it can do its work. And what we see is that when the infected tissues produce these cytokines and chemokines, the neutrophils, normally they're moving in the blood, but they will attach to the endothelium and start to roll on the endothelium, and they become activated, they stop, and then they migrate through the junctions in the endothelial cells. And that's a series of reactions promoted in part by chemokines. So we're looking at it on the bottom in a higher magnification. So here again is our endothelium, which has a whole series of proteins on its surface, of course. Here, the IL-8 is coming through from the infected tissues underneath. That's a chemokine. It binds to a chemokine receptor. That induces signaling, which turns on, in this case, an integrin molecule on the surface of the neutrophil, which binds a molecule on the red blood cell, and that arrests the neutrophil so that it can start transmigrating. So that's what these chemokines do. They attract cells like neutrophils to infected areas and turn on genes that allow them to move there and eventually help clear the infection. And of course, viruses encode countermeasures to chemokines as well. As I said, every step of the way. It's really remarkable. Here are some viral proteins that interrupt the production of cytokines. They can interfere with their synthesis, or they can interfere with the production of functional chemokines by modifying them in some way. Some viral proteins interfere with the action of chemokines. Some viral proteins are actually homologs of cytokines. So if the virus genome encodes a protein that looks like a cytokine, it will bind to a cytokine receptor, but it will not initiate signal transduction, and transcription of downstream genes won't occur. Protein is different enough so that it will bind but not do that. And in the same way, many viruses encode soluble chemokine receptors. They're not membrane bound, they're floating around in the tissues, and they bind chemokines and prevent them to binding receptors. It's pretty evil when you think about it, right? It's amazing what can be done. And also, some viruses can interfere with the signaling. If they haven't blocked the cytokines or the receptors themselves, they can interfere with signaling. Now, I've said lots of nasty things about inflammation, but it's actually good because it stimulates a strong immune response. And viruses that kill cells, what we call cytopathic viruses, they cause inflammation because they're killing cells. The cells are releasing viral proteins and nucleic acids. They're being recognized by dendritic cells, brought to the lymph node, and the dendritic cells are making cytokines as well. So they're activating the innate immune response. And that's why these viruses that are cytopathic, like adenoviruses and herpes viruses and pox viruses, they encode proteins that modulate the immune response, because they're killing the cells that's inducing inflammation because of the viral proteins that are being released and sensed. There are some viruses, however, that do not stimulate inflammation. And these are typically what we call non-cytopathic viruses. They don't kill cells. So in culture in the lab, if you infect cells, they don't break open. And in the animal, they don't kill cells. So cells are not damaged. You don't get any apoptosis. And dendritic cells have a hard time finding them because they're not releasing virus proteins and virus nucleic acids. And so there's very little cytokine production, and there's very little linkage of the innate and the adaptive responses. And so these viruses interact very differently with our immune system than the cytopathic ones. They typically cause persistent infections that last a long time, sometimes your lifetime, because we can't clear them. We can't recognize them and we can't clear them. So inflammation is a good thing. It's a sign of innate activation and communication with the adaptive response. The absence of inflammation is not good. And that can happen when viruses don't kill cells, and eventually we don't know that they're there and we can't clear them. The lesson of all this is that this inflammatory response, which I told you from the Roman originally, heat, swelling, redness, pain, that's the communication of the innate and adaptive responses which we like to have. If we don't have inflammation, if we don't have heat, swelling, redness, and pain, we have an ineffective adaptive response because the sentinels are not interacting with the lymphocytes in the lymph node. And this is one reason, as you'll see when we talk about vaccines, why if we use an inactivated virus vaccine, which can't kill cells by definition, it's not replicating, our immune system doesn't often see it. And so you get poor adaptive responses. So we add adjuvants, which mimic inflammation. We add adjuvants, which are chemicals added to the vaccine. They cause inflammation. Some of them are actually TLR ligands, which engage TLRs and cause the production of cytokines. So the DCs recognize that something foreign is there. Otherwise, with a non-replicating vaccine, they would never see it. As I said before, when you exercise and you have joint pain, that's inflammation that is not caused by infection. There are lots of cases of inflammation where infection is not involved. So if you think inflammation, infection is the same thing, it's not. Get that out of your head. It's two different processes. And here's a great example of that. If you have a skin injury, you've cut yourself, and within minutes, the cut swells and gets red. And if you look on the border, it's swelling and red. That's inflammation, because cutting, just the act of tissue injury, causes the production of cytokines and chemokines by a different pathway than interferon, of course. They recruit immune cells from the blood vessel, as you can see here, and they want to repair it. And so that is inflammation caused by tissue injury. Now, what's really interesting here is that the smallpox vaccine takes advantage of this. Smallpox vaccine is administered using this bifurcated needle. It's a small needle which holds a drop of smallpox vaccine. None of you know this probably because none of you are old enough to have received it, but I get it. They scrape into the surface of your skin with this needle, and the scraping causes skin damage. And that's part of the reason why the vaccine works, because it recruits these immune cells. They've done experiments in animals showing if you inject the vaccine without this scarification, you get a far less effective immune response. So inflammation can be caused by infection, but it can also have many other causes as well. So I want to leave you with this. We've talked today about how infection leads to the sensing of viruses, the generation of cytokines, which attract sentinel cells. They pick up pieces of viral nucleic acids and proteins from the dead and dying cell. They get activated and they move into the lymph node. And there they interact with T and B cells, and we're going to talk about how that works next time. And remember that every step of the way, viruses encode countermeasures to all of these activities and all the ones I haven't mentioned. If they didn't, they would be eliminated. And that includes the sensing, interferon production, interferon signal transduction, cytokine and chemokine action, NK cells, DCs, and complement. There are antagonists at every step. So next time we will continue to talk about defenses, and we'll talk about adaptive immunity, which is tailored to the pathogen.